Hello and welcome. My name is Samarpata Roy, and today I will be taking you through a short lecture on light-driven carbon conversions as part of the MOOC on environmental biotechnology. In previous lectures on carbon conversions, you learned that carbon dioxide or CO2 from the atmosphere is fixed into organic carbon, typically as biomass or related products. This organic carbon can in turn be converted back into CO2, forming a closed carbon cycle. Now, the conversion of CO2 into organic carbon or biomass precursors takes place primarily through photosynthesis. During this process, plants and microorganisms such as microalgae and cyanobacteria harvest light energy from the sun to convert CO2 into organic matter. As we shall see later in this course, it is also possible to convert simpler organic carbon molecules into more complex biomass components through light-driven pathways. Photosynthetic organisms have played a dominant role since the early history of Earth. If we assume that cellular life originated about 4 billion years ago, then by around 3.5 billion years ago, an oxygenic phototrophic bacteria were already active, driving key biological processes on the planet. Over time, oxygen began to accumulate, and we saw the emergence of microalgae and cyanobacteria, both of which use photosynthesis to fix CO2 into organic matter, while simultaneously producing oxygen. You may recall from earlier lectures the metabolic classification of microorganisms, particularly the distinction between chemotrophs and phototrophs. Today, we will focus on phototrophs in more details. These organisms generate energy from light photons, but they also carry out redox reactions using various electron donors. If the electron donor is organic, these organisms are called photoorganotrophs. If it is inorganic, for example, reduced sulfur compounds or water, they are known as photolithotrophs. Depending on the electron acceptor, phototrophs can be classified as either oxygenic, producing oxygen, or anoxygenic, not producing oxygen. Finally, in terms of carbon source, photoheterotrophs use organic carbon, while photoautotrophs use CO2 as their sole carbon source. Let us look a bit closer at photoheterotrophy and photoautotrophy. In both cases, light provides the reducing power, transferring electrons that create a proton motive force, PMF, used to generate energy. However, the carbon source differs. In photoheterotrophy, the carbon source is organic, which is assimilated into cell material. While in photoautotrophy, the carbon source is inorganic, primarily CO2, which is fixed into biomass using electrons from reduced sulfur compounds or water. One of the best forms of photosynthesis is oxygenic photosynthesis, performed by cyanobacteria. Here, light energy is converted into ATP, which powers the reduction of CO2 into biomass using electrons derived from water. This process releases oxygen, hence the term oxygenic. In contrast, anoxygenic phototrophs, such as purple sulfur bacteria, also use light energy to produce ATP and fix CO2 into biomass. But they obtain electrons from reduced sulfur compounds instead of water, so no oxygen is produced. Interestingly, not all phototrophs rely solely on CO2. Some, like purple non-sulfur bacteria, can use organic carbon as both the carbon and electron source, converting it into CO2 and biomass, a process known as anoxygenic photoheterotrophy. When we look at these processes from a stoichiometric point of view, oxygenic photosynthesis essentially uses carbon dioxide and water together with light energy to form organic matter, while releasing oxygen as a byproduct. In the case of anoxygenic photosynthesis, the process still involves carbon dioxide, but the electron donor is different. Instead of water, it might be reduced sulfur compounds, hydrogen gas, or even organic molecules. With the help of light energy, these are converted into carbohydrates, that is, the oxidized donor products and water. But no oxygen is produced in this case. The type of photosystem used to capture light and transfer electron is another key aspect of phototrophic diversity. Despite their structural diversity, photosystems work on a common principle. Light is captured by light harvesting complexes, LH. The energy is transferred to a reaction center, RC, that subsequently reaches an elevated energetic state. The reaction center donates high-energy electrons to an electron transport chain, generating both ATP via the proton motive force and NADPH. Upon losing the electrons to electron transport chain, the reaction center then regains electrons from the electron donor, completing the cycle. Through this mechanism, light energy is effectively converted into biochemical energy in the form of ATP and NADH. The pigments responsible for capturing light are known as chlorophylls or bacteriochlorophyls. Chlorophylls are commonly found in plants, algae, and cyanobacteria, while bacteriochlorophyls occur in purple and green bacteria. Although their core structures are similar, variations in their side chain lead to distinct absorption spectra, allowing different organisms to absorb light at different wavelengths. The graph here compares the relative absorption spectra between purple and green cells, with the green ones showing two peaks for absorbance, that is, at 450 and 680 nanometers, while the purple cells show peaks for absorbance around 800 nanometers. Other accessory pigments, such as carotenoids and phycobilins, show further diversity in light-sensitive pigments that allow for further ecological niches for these organisms. Cumulatively, these differences in pigment compositions create striking diversity in the absorption spectra of phototrophic organisms. For instance, you can see from this graph, cyanobacteria absorb light between 400 to 800 nanometers. Green sulfur bacteria have peaks around 400 to 500 nanometers and again around 700 nanometers. Purple non-sulfur bacteria absorb mainly between 750 to 1000 nanometers. This spectral diversity allows different species to coexist in ecosystems such as layered microbial mats, where each occupies a distinct light niche, with cyanobacteria on the surface and purple and green bacteria in deeper layers. The stratification of these organisms in the mat is, of course, also influenced by factors such as availability of carbon source, electron acceptors, pH, and dissolved oxygen concentrations. In oligotrophic environments such as lakes, rivers, and surface water bodies, a large part of atmospheric CO2 is consumed by cyanobacteria to generate new biomass. Sunlight allows for generation of energy in the form of ATP molecules that are then utilized to reduce CO2 to intracellular storage compounds, such as glycogen. New biomass is formed by utilizing glycogen as carbon source, in addition to fixing nitrogen gas as the nitrogen source to ammonia and utilizing it for biomass growth. In wastewater treatment, we often deal with high organic carbon loads. Ambient CO2 present in the waste in the form of bicarbonate and a wide variety of compounds that need to be valorized. From this perspective, photoautotrophs like microalgae and cyanobacteria are less suited to such environments. And photoheterotrophic metabolism has become particularly relevant. Purple non-sulfur bacteria, a photoheterotroph, excel because of their ability to utilize organic carbon while harnessing light energy. Let us now explore their remarkable metabolic versatility. Purple non-sulfur bacteria, or PNSB, can perform photoheterotrophic conversions using light as their energy source to convert organic matter into biomass. Naturally, some CO2 is produced during this process as well. They can also switch to aerobic respiration, completely oxidizing organic compounds into CO2 and water. Even more fascinating, they can also grow photoautotrophically using carbon dioxide as carbon source, effectively valorizing carbon dioxide emissions from wastewater treatment while simultaneously utilizing organic carbon. In this intricate balancing of intracellular redox mechanisms in PNSB that make it such an interesting candidate for bioprocess applications. This ability to use diverse carbon sources has important metabolic implications. For example, consider 2-carbon substrate, acetate with a degree of reduction of 4 electrons per mole of carbon, and ethanol with 6 electrons per mole of carbon. Biomass typically has a degree of reduction around 4.2. So acetate is close to this value, while ethanol is more reduced, carrying extra electrons. When PNSB grows on ethanol, they must manage these excess electrons. They usually do so in 3 ways: producing hydrogen gas, fixing CO2, or storing electrons in reduced intracellular compounds like polyhydroxyalkanoids, PHAs, which are of growing interest as bioplastics. Thus, their growth not only supports CO2 fixation, but also enables the production of valuable storage molecules. Finally, in a typical diurnal cycle, during the absence of light, these organisms can also adopt a chemoheterotrophic lifestyle or perform aerobic respiration. Because of this versatility, purple non-sulfur bacteria have several biotechnological applications. They can be used for wastewater treatment, biological production and remediation, and nutrient recovery, producing valuable compounds such as bioplastics, PHAs, pigments, carotenoids, and single-cell proteins that can promote plant growth or serve as feed additives.